This is an intro to DNA replication. So in order to have mitosis or meiosis, which are going to be our two types of cell division, DNA must double itself. We want an entire copy of our whole our whole genome in every single cell that we have. So when we want to divide our cells to get more cells, we also need to divide our DNA so that all new cells can have a whole copy. The strands of DNA are complementary to each other, as we know, due to the fact that DNAs have exact pairs. One will have just the opposite of the other, and they run anti-parallel to each other, okay? And we sort of saw this in our other video. If I draw a nucleotide here, the other strand looked like that. So you can see that they run anti-parallel to each other. While one sort of seems to point in this direction, the other points in that direction, okay? And so what actually happens is that one strand can be used to make the other. So one strand can serve as a template for the other strand. So we're going to sort of practice with this. Let's say I'm given this top strand and I need to find the other strand. Well, all I do is write in what the base pair would be. If the template strand says C, my other strand would be G. If my template strand says A, my other strand would be T, and so on. So just given one strand, I can make the other strand. So what I recommend that you do is that you pause the video right now, try to figure out what the template strand will be for the second strand here, and then come back to me in a bit. So why don't you pause now? So hopefully you paused and you're returning to me, and I'm going to give you the answer to this second problem here. The complementary strand for this template strand would be C, A, T, G, G, C, G, A, T, A, T. So hopefully you got that right. I find this to be a pretty easy part of this unit. So heading into DNA replication is semi-conservative, which means that one new strand will be paired with one old strand, the template strand. So this picture shows semi-conservative replication where the blue strands are the template strands, and then the light blue are the new strands. And you can see that in each new double helix, one part of the helix is the old template strand, and the other part of the helix is the new strand.